this chapter is kind of an overview of pulling things together, especially talking about dynamics of how cells are interacting. And I know that we separate as we're covering B cells and T cells. We have specific chapters for B cells, specific chapters for T cells, for innate immunity. But in reality, all of these systems are working together, which is a little ironic because most researchers really focus in on one cell type when they're doing research. Luckily, good institutes tend to have people that collaborate quite a bit. But so this chapter is hopefully going to give you a little bit better idea of kind of the interplay between cells, timings of things, and some of the problems that arise that are techniques that some pathogens will take to try and take advantage of some of these interactions. So again, there are videos associated within. So if you pull this slideshow up, you'll see that there are links to videos, and not all of those links work. So don't get frustrated if they don't all work. Sometimes if they don't work, switch browsers or or open up the link in a different browser. Sometimes that will work. And so I'm going to hopefully try and show you as many of these videos as possible. But again, due to time, please take some time, go through these slides, try and link up. Get on the publisher's website. Go through if you have access, go through the open book on that. You can see you can access videos from that. But they're not vitally important, but they are pretty interesting to see. And it's the actual being able to see the cells interact has actually taught us quite a bit about the interplay of these cells, and so it's really been really vital for researchers. So it is nice to see kind of what has helped us to understand the interplay between these cells. And some of the interplay, watching the interplay between cells has actually brought up a lot more questions in many cases. So again, remember, naive lymphocytes, we have these naive lymphocytes circulating between the secondary. So they aren't static, they're not just sitting around in lymph nodes, they are circulating throughout the system. We have those that are going to the spleen looking for blood-borne pathogens. The thymus is constantly sending out naive cells, they're going to lymph nodes, they're going to tissues. And so by moving through the tissues and the blood, they're able to scan for antigens. They are able to move. So naive cells will have certain chemokine receptors that allow them to traffic from one area to another. Once they've scanned an area, they'll downregulate the receptor and then move on to an area using a different receptor. So they're able to use these receptors to kind of roll. And if you've watched any videos, you'll see lymphocytes kind of rolling either through the circulatory system where they roll along and kind of use their adhesion molecules to help them roll. And then when they find a specific receptor that'll slow them down enough, then they'll pass through those high endothelial venules and into that tissue or that lymph node and be able to then scan that area. Again, they exit the blood through these HEVs, and we talked about kind of HEVs, high endothelial venules. The adhesion molecules, HEVs express L-selectin, or the ligands for L-selectin, and L-selectin is expressed on these naive lymphocytes, and that allows them to kind of contact and slow down as they're rolling along, and be able to pass through these HEVs into lymph nodes or into other tissues. So they tend to, here's just a graphic of them rolling along. Encountering these HEVs, binding to the ligands for L-selectin, which they express, slows them down enough, and they afferent, and you remember afferent because it starts with A, means coming in. So we have our lymphatics coming into the lymph node from different tissues because of that hydrostatic pressure constantly being circulated, and that is going to push through your APCs and antigen. Okay, so that's how your antigen presenting cells are coming in. That's how antigen is being brought into the lymph nodes. But then your lymphocytes, all your lymphocytes are going to come in through the circulation. So we have lots of blood vessels coming through these, and the high endothelial venules within those blood vessels that are passing through the lymph nodes will then slow down. So constantly these naive cells, lymphocytes are circulating. When I say lymphocytes, I mean B cells and T cells mainly, and K cells will circulate, and then when they get to just down the road, they're getting down the road, they pass by that lymph node, they say, okay, I'm going to slow down, enter this lymph node. They're going to hang out in that lymph node for a day or so. They don't find antigen, they'll downregulate the chemokine receptors that keep them in there, like CXCR5, that keeps them in the T cells will downregulate that CXCR5 that keeps them in that T cell zone, and then they will enter back in or exit if they become activated, they'll exit through the efferent. If they don't become activated, they will then head back out into the bloodstream, go down to the next lymph node, and then start all over again.
When they, always, which one, you said NK cell, lymph node, whatever B cell, they come through the blood. So which one are the one that through the HEB?
They both go through an HEB. Both B cells and T cells both go through HEBs. But then the area that they go to will express, like for example, T cells will express the ligand for they express CXCR5, which the ligand for that is going to be in the T cell areas and in your these areas in here. And then the B cells will then be attracted to the chemokines that are in these follicles. So you're going to get mainly B cells in the follicles, but they're all coming in through the bloodstream through these HEBs. T cells will tend to they will traffic to the T cell zones, and they will move along these basically these pathways, fibrous pathways that are formed that will be lined with dendritic cells, and they'll scan for antigen. And the B cells will move along the basically these dendritic cells that will form these same types of pathways.
Dendritic cells, they don't move, they stay there.
These dendritic cells stay there. Now there are resident dendritic cells, and then there's dendritic cells that will come in.
Blood. So if dendritic cell come from blood circulation, where do they come in? Is also through the
dendritic cells aren't coming. Yeah, the dendritic cells, as I said, so APCs include dendritic cells, so they'll come in through the afferent, through the lymph.
So even with the lymphocyte, right?
Yeah. So mostly it's just lymphocytes coming in through the bloodstream. And if you think about it, lymphocytes are much smaller. They can roll around through the bloodstream. Dendritic cells, very large, will tend to come in.
So how do they go, or where do they go, dendritic cells? How do they go to the?
Through the afferent lymphatics?
Yeah.
Yep. So usually you'll find dendritic cells in tissues, and then they'll traffic along into the lymphatic system and move through that system.
So what's the function of HEB?
HEBs are kind of an entry point for T cells and B cells for lymphocytes, and they also express the ligand for L-selectin that slows them down and allows them to enter.
Slow down. Yeah. So it's entry and then slow down, like kind of come on to search for something you want, right?
Yep. So here we see in these two videos, I don't believe work. Unfortunately, I think they're dead links. I can try them again now that I made my default browser Firefox. And I'm not even sure which of these videos is the video they think it is, which I lymphocytes moving along these, so we have our green T cells and red B cells, and we have areas that are, as you can see, are B cell rich in B cells, and areas that are rich in T cells. And then we have, in this middle screen here, you can see T cells, which are your green, interacting with the fibroblastic reticular cells, the FRC, remember? That's your fibroblast reticular cell network in red, and we can see that these cells, these T cells, if you saw the movie, would be rolling along, moving along, following the path of these FRCs.
What's the function of FRC? Can you say that?
The FRC gives so if the T cells came in and you didn't have the FRC, they wouldn't have
They cannot roll.
there would be no pathway. It's kind of like giving you a path and saying, you know what, you go through this path, you scan this path for antigens. It helps to focus antigens in, and so you have the ability to kind of have a network of antigen presenting cells, of antigens that will then be scanned by these. So basically it creates a network that allows the T cells to interact with the antigen presenting cells. And if you didn't have that, it would be harder for them to get together, scan each other, and then move on to the next one. It would be more random. And this allows it to be less random and a little bit more of a structured interaction.
So it's just like a liaison, like a communication between
the FRCs will move along, the T cells move along the FRC because it has adhesion molecules that they will now bind to and roll along. Yep. And cytokine receptors, or chemokine receptors, sorry. And then here in this last picture, we can see that the T cell, which is going to be your blue or your cyan, is interacting tightly with that FRC, follicular reticular cells, our fibroblast reticular cells, and you see kind of that they are very closely associated. We'll scan all these cells. Again, chemokine binding to chemokine receptors account for the different migration of cells. So the FRCs are in the T cell zone, express CCL21. So your question is, how do they know? It's because the FRCs are expressing CCL21 and CCL19. That attracts naive CXCR7 positive T cells. And then the follicular dendritic cell network that's in the B cell zones expresses CXCL13, and that attracts CXCR5 positive B cells. So, and then remember how when B cells encounter antigen, they'll express both CXCR5 and CXCR7 and localize to that interzone in between the T cell zone and the B cell zone. So,
what's the last word you say?
So you have your T cell zones, and T cells will localize there because they express CXCR7. That binds to CCL21 and 19 in the T cell zone. That's how you differentiate the T cell zone with the B cell zone. But then when B cells, they'll localize to the B cell zone because of CXCR5, but then they'll upregulate CXCR7 when they find antigen. But they'll still express CXCR5, and so they'll kind of be torn between the two areas, and that will basically localize them to that interface between both regions. And that's where, and why would they want to do that? Because they want to now get help from CD4s. So activated B cells will not continue to proliferate. They won't go on to create germinal centers. They won't go on to undergo somatic hypermutation. They won't do any of that unless they get help from a T cell. So where are they going to get help from a T cell? In the T cell zone. So then they go to the T cell zone, and T cells, as they're moving along in between these two zones, will then scan the B cells. And if a T cell finds that B cell that is presenting an antigen that it is specific for, then it will provide that help. That B cell then will go, and it will either become a plasma cell, or it will enter into a germinal center and continue to become more antigen specific in the
common plasma cell, they produce a secreted antibody, then go to the germinal center to have mutated.
And those can then become plasma cells as well. So you have plasma cells localizing, they'll exit through the efferent.
Yeah.
They'll go to the medulla and then they exit through the efferent. They'll go to bone marrow. They'll go to sites of inflammation. Go to tissue. Go to other secondary lymph or tertiary lymphoid organs. But then you have also
So what's a long-lived plasma cell in the bone marrow?
Well, that would be a memory cell. That would become a memory cell.
So they also call it plasma cell?
They're plasma cells. They're
It's
large and
confusing.
secreting antibodies.
Huh?
Yeah.
They can also secrete, because they said that, okay, after germinal center, they can become plasma cell again, then move to the bone marrow.
Yeah.
So they didn't say that's memory, they have memory, how do I know which plasma cell they go through? Is it beginning, before they go to germinal center, they have plasma cell?
If you could answer that question, you'd probably have your own lab and get a big grant and make lots of money. So yeah, that's a question a lot of people are asking is, how do I know? And it doesn't matter, is there a difference? So that's one of those questions that, and that's why immunologists are excited about imaging, to be able to follow the path of cells. Now we need markers that will tell us whether that cell, and we use things, we do have tools to see if the cell's divided, how many times it's divided once it's been activated, but they're not very long-term tools. So yeah, I mean, it's one of those questions that a lot of people want to know, but it's one of those questions that isn't really easily answered.
If that's I'm confused, that's why I asked it. Because if that activate the B cell, kind of, go to other route, go to whatever, it's called a primary site. What's a primary site or something?
A primary site.
Principal, whatever. Okay, they secrete, the first, before they go to germinal center, they become activated, secrete antibody. Where do they go? Medullary, right? Medullary.
Yeah, and then they'll exit through the efferent and go to sites of inflammation, or they'll start secreting. They'll stick around and they'll secrete antibodies into the bloodstream.
Do they go to bone marrow too?
They can go to the bone marrow. They can be secreting antibodies there as well. Continue to secrete antibodies which enter the circulation. So yeah, but a lot of these are very systemic responses. Antibody responses can be fairly systemic, which means that they're going to localize to certain areas. Those areas are going to provide. Why would they localize to the bone marrow? Well, bone marrow has other factors, other chemokines that'll secrete that'll allow for cells to continue to have survival signals and continue to kind of become a little more long-lived. They'll secrete antibodies. It doesn't really. Sometimes those antibodies don't have to be local. If you have a blood-borne pathogen involved, I mean, it's going to be throughout your entire system. And so that'll secrete antibodies, they'll circulate in the entire system throughout the whole body. So you can find those antibodies in any tissue, or, if you draw blood, obviously that's blood that's been circulating through the whole body over and over again.
Yeah, it's confusing is when whatever stimulation is gone, whatever inflammation is gone, all of the antibody secreting stuff is, turned down.
Yeah.
So is those plasma cell before the germinal center one?
Well, some of them will some of them will apoptose, and some of them will become memory cells.
So they can be.
Yeah, we don't know what deciding factor makes them decide I'm going to apoptose or I'm going to become a memory cell. There's still a lot of debate about what is the trigger for that. Is it just stochastic in that some cells do and some cells don't? We don't really know.
So the germinal center one has a higher affinity for the specific antigen, plasma cell?
Well, what do you think?
I think so, but how do you know?
Well, because those are going to undergo somatic hypermutation. So you can tell that undergone that because they're going to class switch, right? Because class switching is also involved in the germinal centers, and they don't class switch if they haven't. So you would have IgM would be the major secreted antibody for cells that don't go into germinal centers, right? So you can kind of tell the difference.
IG other.
So you would, if it comes out of a germinal center, chances are you're going to have IgG, you're going to have A, E, whatever, you know. But you will see that you could take those cells, clone them, put them in a dish, expand them so you get enough DNA to work with, and look at that DNA. The DNA is a clone of every single one of those. You look at the DNA. You look what's been cut out. I can sequence that DNA now. I can look and see what's going on, and then I can tell what happened to that cell. But again, it's hard to tell things in vivo because I have to take the cell out, I have to expand it, I have to kill it, I have to stop and look in this point in time. And that's kind of the point to this chapter is a lot of what we knew before we could start imaging cells was snapshots in time. And it's hard, I guess, to give the example of a sport. If you were to try and learn a sport like football or something, by just looking at pictures of that sport, you know, it'd be really hard to understand the dynamics of that game if I'm just looking at snapshots of the field and players on the field. I might see that there's players with different colored jerseys. I might see that there's different size players or something. I might see that the players, localized to different areas at different times, and I have to put together how is this game played. The same issues going on here with the immune system. We're looking at snapshots. When we draw blood, we're drawing it at a specific time. We're looking at cells that localize to the blood. If I want to look in the lymph node, I'm going to have to excise a lymph node. So either I'm going to have a patient that's willing to give me a lymph node, or I'm going to have to work with a mouse who's maybe a little more willing to give me a lymph node. Or I might have to, and then I'm still looking at a moment in time at that lymph node. So which is why it's so great to be able to use some of these visual techniques where we have fluorescent proteins that we can now drive. So here, in most of these cases, we genetically modified cells by putting in a virus that expressed GFP on a promoter for a T cell promoter, or I have a promoter that only expresses a protein in a dendritic cell, and so I'll drive GFP on that. I'll drive a red fluorescent protein on another, and so now we can kind of start to watch these cells interacting with, and which is the case for a lot of these. Or I can stain these cells. I can put an antibody that's specific for those cells that has a fluorochrome, that has a fluorescent antibody on it, or a fluorescent tag on it, and then I can watch those cells. So that's kind of the point to this chapter, is to see what we've been doing research-wise, and how we know what we know is about the interactions of these different cells. So let me pause that. That was an actual movie that worked for once.
So some grains out of the
So for this, the
T cells.
Yep. So you've got T cells zone in the paracortex. So that's in this PC paracortex area. The T cell zone. You have your let's see. The T cells in this case are red, and then the B cells are blue, and then so let's see. Go back to So T cells in this case are red, and the B cells are blue, and who's green? So it's actually, it looks like in that video we're looking at, I'll tell you right now, in this case, okay, so here the dendritic cells are green. And there's macrophages that are red in the parrot. And then let's see, for B, so this is actually A. So we're looking at kind of dendritic cells Yeah.
All right around macrophage?
Yeah.
Are you sure?
In that video, right, there we go.
Yeah,
I
and so the
see.
macrophage tended to kind of sit and present cells and be kind of a background. Let's see, they only have that one video. Okay, so here you can see in picture B, we are looking at dendritic cells are mingled with T cells. T cells are red. Again, dendritic cells are green. T cells are red. And this is in the follicle. And then in this you can see that the B cells are blue, and dendritic cells are green. So here we're seeing dendritic. So we're focusing in on the movement of dendritic cells in these cases. Dendritic cells are constantly coming in to the lymph node and exchanging antigens. Again, dendritic cells, macrophages may be presenting antigens or may bind to antigens. Macrophages also have your PRRs, so they can sense when there's pathogens around, and they can pick up certain certain molecules associated with pathogens, and then they can pass those off to dendritic cells. Dendritic cells will come in, or the dendritic cells will come in already activated, already bound to antigen, and come in and kind of then they will populate areas where T cells can now scan along those dendritic cells, and the same in B cell areas. They will then present antigens to B cells, and so you can kind of see in these pictures, in these snapshots, that the T cells are pretty tightly associated with the dendritic cells in a lot of these cases. And the other cases, if we were able to look at the video, you'd see a lot of these that aren't associated or kind of rolling along, and then they will become associated depending on if those dendritic cells present their specific antigen. So T cells and or B cells will come along, and they won't spend a lot of time on that dendritic cell unless it happens to have an antigen that binds to its receptor. Then it'll slow down and have this period where it's then not tightly bound. It can bind to other parts of that dendritic cell, but then it has up to an 8-hour period where it now will bind to that dendritic cell, and they will communicate. They will form a union, and then that T cell or B cell will become activated.
So if I understand correctly, the most activated dendritic activity is in a T cell form.
Based on the picture, but I wouldn't draw that assumption just based on those pictures. Again, those snapshots are area just dendritic cells. So those are dendritic cells that are entering that we're looking at. There are also resident dendritic cells that make up the scaffolding on which B cells will circulate. And if you remember, you can see those little beads of antigen just sitting on the dendrites of the dendritic cells. And those are kind of what B cells are going around scanning. So the B cell area is actually very rich in dendritic cells as well. So you can't, B cells, but there are resident dendritic cells. There are also dendritic cells that are coming in and circulating in and out, bringing antigen with them.
Which one is more is resident or circulating dendritic cell?
Don't know that. Yeah. So it's
The resident one is the one has a kind of beads, right?
yeah, but they can trade out. They can swap those with other dendritic cells coming in. It's very permissive, which is why it's hard for me to say one's more important than the other. Sometimes tissue resident dendritic cells are really important because they are the first responders to an insult from a pathogen. So a pathogen comes into my skin. I have Langerhans cells that are sitting in layers of my in my dermis, and they will then they have PRRs on them. They'll see they'll start to phagocytose everything in the area, and then they'll start to traffic. They'll then traffic through the circulatory system, move to the nearest lymph node. And then as they enter the lymph node, as we saw, you get those green dendritic cells you saw kind of coming in and out and kind of probing around, probably had been stimulated, activated, and in tissue. In this case, it was an experimental system, so they probably had injected an antigen, or they probably injected an antigen subcutaneously or something like that. Those Langerhans cells became activated, entered into the to the lymphatic system, entered the nearest lymph node, and started presenting antigen to the T cells and B cells in that lymph node, saying, hey, something's wrong. Do any of you guys see something out of place on me? They start to probe those dendritic cells, and then the T cells and B cells now start to do something. So if the lymphocytes fail to find antigen in the lymph nodes, they upregulate S1P1 receptor, and this allows them to leave. So again, they're going to downregulate the receptors that hold them in that area. You continue to allow them to circulate in there. And they'll leave through the medullary sinus, and then these cells leave via portals to return to the circulation. Image shown, it shows the tracking of lymphocytes as they exit. So here they're kind of showing the path, the little dots they're showing. Some poor postdocs sat and watched each of these cells as they passed through the field of view, drew little dots showing the path, and then showed that as they moved through, then they would exit the lymphatic system. So you can see that as they're not being stimulated, in this case, they didn't provide the cognate antigen. They probably had OT1 cells that were specific for ova, and they didn't provide ova, and so the mice don't make ova, so if they don't have their antigen, those cells are probably just circulating through lymphocytes, don't see their antigen, exit the lymph, exit lymphocyte, but the lymph node, they enter lymph node, exit the lymph node, and you can kind of watch that happen, just as they did show in this video. The innate immune, other cells, now we talk a lot about B cells and T cells and dendritic cells, but there's, again, remember some of those innate cells, and a lot of these immune responses are going to rely on, even though we have adaptive responses to bacteria that are really important, sometimes to get those interactions going, we initially have innate cells, like macrophages, neutrophils, that will have these PRRs, these receptors that are specific for pathogenic molecules, and they're activated by PAMPs, pathogen molecular protein associated, pathogen-associated molecular proteins or products, and then also DAMPs. We don't say DAMPs up there, but damage-associated as well. So and we evolved to have those damage-associated interactions as well, because maybe we won't recognize anything specific on a pathogen, but it's really tearing things up. It's eating my skin, or it's doing something, and that damage is another signal. So that's why sometimes you can have a cut, meaning you clean it out, you'll still have some inflammation in the area. Doesn't necessarily mean that it's infected, but damage can cause an inflammatory response as well. And this induces intracellular signals that coordinate killing of pathogens. They alert adaptive immunity to the infection. So again, like the example of skin, you have dendritic cells, and in this case, dendritic cells don't have a specific receptor. They don't have T cell receptors. They don't have B cell receptors. But they do have receptors that will recognize some pathogen-associated molecules, and then that will activate that dendritic cell to, instead of just sitting there, will now start to phagocytose things. It will start, and then neutrophils will come in, and here's what they granulocytes are what we're watching a video of. So they, in this case, we see the neutrophils, which are green, and then we see these swarming protozoal parasites, which are red, and we can watch, hopefully, we can watch this interaction. So these neutrophils are not antigen specific. They don't have B cell or T cell receptors, but because these pathogens, these protozoa, have markers that are associated with the receptors, so they have a common pathogen-associated molecule on their surface. These neutrophils are coming in, and they're just swarming in, and they're degranulating and creating a pussy environment. They're creating an inflammatory environment, and they move to the area of infection.
So, because they can not live because they are this protozoa or whatever parasite, so they are not inside the cell, so they cannot kill, they cannot internalize. What they can do is...
Well, a lot of times the neutrophils, very large cells, and some of the macrophages, they will internalize some of those protozoa, and as we'll see in the case a couple slides down. Sometimes they will swallow, and in those cases, they'll either, if they're effective enough, they'll digest them, and if they're not, then those will become an intracellular pathogen. And there are a lot of intracellular protozoal pathogens.
So it's hard to kill those
Yeah.
protozoa, right? So
It is, yeah. Which is like, it's kind of lame. So here's, then we'll talk about that in a second. So let's move on really quick. Antigen presenting cells travel to the lymph nodes. So antigen presenting cells, let's say in that circumstance, some dendritic cells or macrophages took up some of those pathogens. They moved to the lymph nodes and presented, they processed the antigen, they presented it to T cells, and once alerted to the pathogen, so the ABCs migrate to it. So again, we have a lot of APCs all throughout, they circulate throughout the body, they are in the tissues, and once they are alerted, they'll then traffic the lymph nodes and present their antigen to the lymphocytes there. And again, they enter through these afferent lymphatics. And this is where then they can come in, and they will also associate with these FRC networks. In the T cell zone, for example, they'll come in and they will bind to these cells. And this is where you can get these complexes where CD8s and CD4s both bind to dendritic cells in these areas. And again, cross-presentation is necessary to really drive a good immune response. And here we can, whole antigens can also migrate to the lymph nodes, so not just dendritic cells, but antigens themselves. Again, remember, we're constantly flushing fluids through our tissues, moving it through the lymphatics. And also, antigens can arrive intact from the blood, through the spleen. These antigens can arrive opsonized. So again, remember, there's this complement proteins, there's antibodies that will bind to things, bind to complement. So the antibodies might circulate, bind to those things. They can come in opsonized by complement and or delivered by specialized macrophages. So all of these are possible routes of entrance for antigens. And here, the picture we're going to see, hopefully, we'll even just look at the picture, but here you can see that they have antigen, in this case, entering, and these guys are deficient in complement receptors, and these guys have complement receptors. So in this case, you can see that whole antigen will come in, and it will associate with strip away the antigen from macrophages, but in this case they can't. So,
What is this with?
so let's see, this one, green, the B cells are green in this case, and the macrophages are red, and the macrophage that has this antigen is red. So the antigen is bound to these macrophages, and then if they have complement on them, B cells will come along and they'll bind to the antigen, they'll bind to the complement itself through CD21, and then they will be able to internalize that antigen, process it, and present it via MHC class 2. And if they don't have those receptors, then they won't. So here they're not able to grab the antigen away from the macrophages and bring it into the B cell zone. So here you're kind of seeing in the case of, let's say a macrophage came into the area, or the resident macrophage bound to this complement, to this antigen that's bound to complement, bound to antibody. And it kind of sits there on the surface. Now B cells will come along, they'll bind to it, bring it into the B cell zone. Dendritic cells will also have receptors for this complement. They'll then present that to other B cells. So it allows for kind of the spreading of this antigen and kind of the processing and movement of antigen from one area to another. That in this case is driven by complement bound to that antigen. And so if we don't have, so we can see the role that plays if this video plays. But if not, you can see you can see that the B cells in these cases are spending a little bit more time associated with the antigen that's bound to macrophages, and then you can see in some of these cases, you can see some of the red looks like it's running down, it's getting together. You can see B cells that are leaving bound, and they're kind of spreading that antigen. So this antigen is staying associated with macrophages in this case. But in this case, that antigen is kind of being spread down into the B cell areas and presented. And you can see kind of the movement of the red from this area down to this area as B cells then will come in, they'll grab the antigen that's bound to complement, they'll now bring it into the B cell zone, and then you see that red then disassociating with the B cell because it's now binding to the dendritic cell that's resident within that B cell zone, and it will now present that to other B cells. So it's kind of just, this is to show you that, again, this is all very dynamic. It's reliant on lots of factors, and it's reliant on complement working, it's reliant on antibodies binding to this antigen, and binding to complement. So, and without watching stuff like this, without being able to see this interaction, it's harder for us to, with that, just that snapshot, to say what's going on and how the antigen got. So not watching that, you would say, well, maybe the antigen just leaked into the area. How does it do that? We don't have, there's not really good flow of fluid from the areas, but maybe there is. It's really hard to watch something like that in an organ. But by watching that video, you can see that, oh, B cells are actually actively going, grabbing antigen, bringing it into the B cell zone. It's actually, they're probably competing with dendritic cells, it's binding to dendritic cells. So you're able to see, and so in that same vein, if you have a deficiency in the receptor for complement on dendritic cells in particular, or on B cells, or systematically, you're going to know that, oh, this person may have a deficiency in being able to present certain antigens within their lymph nodes, and therefore they may be immunosuppressed for that reason. So, yeah. So naive CD4 T cells, arrest their movement after engaging antigen, and here we can watch here T cells in red bind to dendritic cells in green and slow down as they bind to their particular antigen. So you can kind of see that here's an antigen-specific T cell coming up, and once it's come in contact, now it's stuck. You have other T cells that aren't necessarily specific, just kind of moving around, but that's
How do you know they slow down?
because they've watched millions of. We get to see the one good slide. They watch lots and lots of slides, and these guys then will actually stay bound like this for up to 8 hours. And
so
this is just showing us 3 minutes of that interaction sped up.
So they just sit somewhere and not move?
So that they will sit bound to each other, and then for 8
So
hours.
they colloquize?
Mhm.
How do they know, that's one? So they move
Because
around.
their T cell receptor will bind to that MHC, presenting the antigen. It will then create that C-SMAC and P-SMAC, and then that will stay maintained, that interaction, all those adhesion molecules, they'll stay bound to each other. They'll be processing signals, T cell receptor signals, will be sending signals to the T cell. Cytokines are going to be secreted locally, taken up by that T cell. That T cell is then going to decide, do I want to become a Th1 cell, a Th2 cell? All that's happening in that 8-hour period. And then once that T cell dissociates, then it will start to proliferate.
So
they call that a long-term relationship.
So they just do that.
So for T cells, a long-term relationship is 8 hours. So the kinetics of that interaction, for example, and this is just based on conjecture, based on watching some of these pictures. We believe that a lot of times T cells will come along, they'll slow down, maybe they'll bind to a different part of the cells. So stage 2, that you have this transient interaction where a T cell will move, it'll slow down, it'll move from dendrite to dendrite, and then you'll get these 3 to 16-hour clusters of T cells that will then associate and very tightly with that dendritic cell. These are all antigen-specific cells, and based on how long those cells stay bound, it will then directly correlate oftentimes with how much they proliferate and how active they become, or how inactive they become, depending on the length, the association of that interaction. So after binding antigen, for example, we saw T cells after binding antigen, B cells upregulate CXCR7. Again, that brings them into the T cell area, kind of interfacing between the B cell area, because they express CCR5. They also express CCR7, which brings them to the T cell area, and then that allows them to now associate with T cells. Image shows the antigen-specific B cell in green and the non-responsive B cell in red. And here we see, so in this case, they have B cells that have expressed a certain receptor, and they will give the antigen for that receptor, but they also have B cells that are red that don't have that receptor. And so we should see, based on the theory, that activated B cells will co-localize to this border region. We should see more green than red cells at that region, based on that hypothesis. So here's
So the brain is what?
So here they're showing kind of this area here is going to be that interface So even if you look at some of these red ones, they're still kind of moving around. It looks like they're just kind of stuck in a certain area. They're not, but then they moved out of that area. This guy, he did pass through and kind of moving out. He's there. You're thinking, oh, he's gonna stay there. Probably not. He's gonna move out. So he's moved away. So if you watch a lot of these, the majority of those cells, again, in this area are green because those are the antigen-specific ones. They're the ones that have seen the antigen, and they're expressing now both CXCR5 and CCR5 and CCR7, and they localize to this region in between the B cell area and T cell area. Okay, so
what's the area called is TB cells?
the interface between the T cell and B cell zone.
What's the name from it?
T cell interface. So here we see this is going to show activated B cells in green, and then your activated helper T cell in red, and this is going to now depict that our belief that the reason why the B cells are going to localize to the zones between B cell and T cells, because then you're going to get them, and you can see they're kind of bound to each other, following each other around. Here you can see there's a B cell and a T cell, and these guys are even though they're moving around, they're tightly interacting, they're communicating with each other. That T cell is providing help to that B cell. So very tight associations between the T and the B cell. And then here, then B cells, once they receive help.
There's a T is a T4.
Yep, those are helping the CD4s. And they said, because they said that it's very likely that just CD4s are red. So they either labeled them with the anti-CD4 red, or they drove red expression on a CD4 promoter. So then here you have dynamic imaging using looking at B cells, activated B cells in germinal centers, and here's where you get actually more questions than answers, because when they looked at this, they were saying, well, we associated dark zones with cells that were undergoing hypersomatic mutation, and then the lighter zones we associated with cells that we were becoming plasma cells. But when we actually followed the cells, looking at these higher affinity cells, we can see the higher affinity cells that are actually they are moving to these areas, but then they're moving back into the dark zone areas. So it actually tracking experiments show B cells move only rarely between the two zones, and that, again, as it says here, raises more questions, because we're teaching you kind of one thing, and then we're seeing sort of interactions to say, well, maybe that's not how it is done. We're not 100% sure.
So we don't know.
We don't know.
So, but you do know is dark zone is similar to mutation, right?
Yeah. We do know that that's kind of where that's starting, but we
The lighter zone, what kind of cells in a lighter zone?
you have a little bit of everything. You have apoptotic cells. You see more apoptotic cells in that light zone, which is probably what creates the lightness of the light zone. But as far as cells that have undergone hypersomatic mutation, they kind of will move very rarely in between the two zones.
So FDC is where?
The FDCs are throughout the follicle, but then you get germinal centers that form, and they don't require FDCs.
Or they don't need a FDC?
Yeah. So
Lighter zone do have FDC.
I mean, there is that scaffolding that kind of threw out there in the B cells, but majorly what you have is a lot of proliferation going on. They do have cells that represent antigen, because you do need them for selection again, but that answer I won't be asking that question. So. CD8 T cells are activated activated in the lymph nodes. So now we looked at B cell activation, they needed CD4s. Here now, how do we get CD8 T cells activation? And here we can look at the motion and arrest of a CD8 T cell when they recognize and present an antigen, even at low concentrations. And here we'll just look at the pictures, and we can see that when you don't provide peptides, the CD8s are kind of buzzing through there. Very low levels of antigen you can add, and it will slow down CD8s that will start to now recognize it on antigen-presenting cells, and then you add even more, and it slows them down even more. So we're not going to watch that video because we're running out of time. This video is cool because we've always conjectured that how do CD8s become activated? How do they get help? And we've always said that, well, it's possible that they co-localize on the same dendritic cell as a helper cell, but then actually being able to visualize it happening really confirms that it does happen. So here we can see that in this case, so it happened really quick. Here we have a CD4, a helper cell, associated with a dendritic cell. And that helper cell, obviously, again, remember, helper cell sent a signal to the dendritic cell to say, hey, start cross-presenting your antigen. So that saw an antigen via MHC class 2. It activated the dendritic cell to now open up the MHC class 1 pathway to exogenous antigens. And it started presenting exogenous antigens via MHC class 1. And you can see the CD8's coming up, and he's just looking around. And he's going to see his cognate antigen, MHC, on this dendritic cell, and then he is going to quickly now associate. And here you have both the CD4 and CD8 are bound to that same dendritic cell, and now you have cross-presentation. You have CD4 providing help. CD8 is getting activated because it sees its cognate antigen. It's also receiving co-stimulatory molecules from the dendritic cell, and it's receiving help from the CD4 cell. So all those factors are in place, and now that CD8 can become a cytotoxic T cell. And then these cells, once they're activated, will exit the lymph nodes and enter back into the circulation. And as some of the pictures we saw previously, we can see that they'll localize to all areas. Plasma cells, so B cells will then early IgM producers move to the medulla of the lymph node. IgG producers often move to the bone marrow. IgA producers localize to the MALT and the gut. And then you get T cells that will then travel to multiple organs. And when we looked at activated T cells in the mouse, remember, we didn't see as many. A couple days after antigen was introduced, you didn't see a whole lot of them in the lymph nodes, but you saw them in every single organ. And so they will then follow different chemokine cues to send them to different areas. And here's just a review of kind of what we said. You have early hours of APC activation. You have antigen-presenting cells coming in. You have your lymphocytes coming in through the HEBs. They will now then, so first you get antigen enters, you get your lymphocytes come in and start looking for cells bound to that antigen. T cell APC interactions occur 1 to 24 hours after infection. 24 to 96 hours after infection, T cells start proliferating and differentiating. You get swollen lymph nodes. You start to feel sick. So a lot of times that feeling is sick. So you saw your buddy on the weekend. He was sneezing, red eyes, runny nose. Come Wednesday, Thursday, now you start to feel it. Your lymph nodes are swollen, and you are also infected. You probably got infected on Saturday, but you didn't really start feeling it until your lymph nodes started swelling. You have chemokines rushing through, cytokines and causing inflammation. You have lymphocytes now trafficking to different organs in your body. So a lot of things are happening. That's where a lot of that soreness comes. You also have your B cells. Again, same thing is happening with your T cells. Same thing's happening with your B cells. B cells migrate to T cell zones if they see antigen. again, we saw the picture of B cells actually looking for, grabbing antigens away from macrophages and pulling them into the B cell zones, like dendritic cells are taking up that antigen and presenting them to other B cells. Those B cells got activated, they're looking for help, they receive help, and then they go and they start to proliferate and move, create terminal centers, and you get antibody production and move out into the, and they all go out through the efferent lymphatics and start to cause all that trouble or help, however you want to look at it. And then 10, 14 days later, which is why they say if you're still feeling sick after 2 weeks, come and see me because you probably haven't cleared that infection. But hopefully 14 days later you're feeling better. Antigens removed, most lymphocytes are no longer proliferating, most of them are starting to die, or if not already dead. Again, remember T cells, once they're activated, will start to express fast. All T cells express fast ligands, so they'll actually start to inhibit each other and start to kill each other. Regulatory T cells may also be induced over time. The more and more you see the antigen, the more regulatory T cells that are induced. Remember, we have these inducible Tregs that are in the periphery are eventually induced and will help to suppress that immune response. Which is why sometimes if you have a chronic response that lasts longer, it will have outlived your primary response, and then it will become a chronic issue. Different tissues have different chemokines, and chemokine receptors on different cells will direct them to different tissues. So even though you do find lymphocytes in all the tissues, it isn't always as random as it may seem. And here we see the case of the parasite T. gondii, or Toxoplasma gondii. This is the one that is found in cat feces or cat urine and feces, and creates spores. And this is why you shouldn't change your litter box if you're pregnant, because actually 50% of people have this parasite. And it doesn't really cause any, there's no phenotypic symptoms for it. In general, we keep it in control, but then if you're immune suppressed or if you're pregnant, this parasite can really affect the fetus, and so that's why you don't want to change the litter box. Wash your hands as often as possible. They also say that if you clean the litter box every day, that you don't prevent lower those numbers, but whatever. So a lot of people have this parasite, and in this case what happens is that parasite will actually enter into areas in lymph nodes or the spleen, and it will be taken up by neutrophils or macrophages, in this case, and which is a normal response, because in the case of like virus, for example, you want macrophages that will bind to that virus, and then they will actually bring that virus into areas where that virus can then be processed and by antigen-presenting cells, and you can start to get rid of it. But in the case of some toxins like this Toxoplasma gondii, or not toxins, but parasites, this will actually infect macrophages and move into areas where T cells are, and then actually infect T cells as well. So they're pretty hardy, and they're able to kind of actually, and in this case, you can see macrophages actually play a role as a vector in moving, because the macrophages will circulate throughout the system in an attempt to alert the immune system, but in this case, they actually are a vector for passing on this parasite. So there are some cases where this incredible, wonderful immune system of ours can be hijacked, and this is one of those cases. Here's an example of when you took anatomy and physiology. You probably heard of this blood-brain barrier, and most lymphocytes will not traffic through the blood to the brain, but here's an actual case that imaging really taught us something very interesting, that these T. gondii that get into the brain, they kind of have evolved to go to localize to the brain to cause mice or rodents to not be so turned off by the smell of cat urine, and causes them to actually behave in a way that makes them more susceptible to getting eaten by cats, so that that pathogen could then be, because apparently this pathogen will only reproduce in cats. And so it turns them into little, it makes them love cats. And some people have theorized that actual cat lovers may be infected with T. gondii, and therefore they love their cats more. So it can affect mammals to make us a little bit crazy. But so it localizes the brain, and how do we respond with this? We actually can have T cells enter the brain, which is something that before now we really didn't think happened a whole lot. But then we saw that these pathways were formed within the brain. These fibrous networks actually were established in the brain, and here we can see these blue fibrous networks along which T cells, just very similar to the FRC networks that are in lymph nodes, were actually formed around pathogens in the brain and allowed for the T cells to kind of move along this network just like they would inside a lymph node and localize to the pathogen and kill the parasite, kill them, or phagocytose them, or become infected, whichever is the case. We've also learned a lot about trafficking and presentation of antigens with skin grafts, using skin grafts. Looks like we're out of time. So Wednesday, I'll finish this chapter up and move on to the next chapter.
We don't have Wednesday. Wednesday is holiday.
Oh, Wednesday's holiday, so no, I won't cover this. Why don't you guys, on your day off, just go through the slides, lots of videos. Again, as I said, I'm not going to have a whole lot of questions from this chapter because it's really hard to show a movie in the exam and ask you questions about it. But again, I do enjoy the last part of these slides are actually looking at some cases of what's happening actually in the site of infection. In this case, we learned a lot from skin grafts, knowing that when you have a skin graft, you have dendritic cells that move in that skin graft, will then grab antigen, move to lymph nodes, and then present that antigen. And so we know this because we can take skin grafts and we can manipulate the cells that are in them, and we can manipulate the cells that are in the mouse that we're putting the skin graft on, and we can do some kind of play around with that a little bit. But you can go through and look at these slides. Also look at how listeria, we had some really bad listeria infections in 2011 associated with cantaloupes. You can see how listeria hijacks the cells pathways to actually launch listeria into the next cell and dendritic cells, and kind of look at T-cell tumor responses. T-cells are also involved in removing tumor. We know that, well, for many reasons, but one is that people who don't have proper T-cell responses have an increased incidence of cancer. And we know that a lot of times you'll see cancers regress, and a lot of those regressions are due to the proper immune response being mounted naturally, and that will happen without even giving chemotherapy, which is why a lot of people are trying to keep that in mind when they're giving chemotherapies, and that we want to give chemotherapies that are going to wipe out the tumor, not the immune system, because a lot of times the immune system is keeping that tumor in check. And if we wipe out the immune system along with that chemotherapy now, in some cases, that cancer can thrive and grow out of control. So it's something. But looking at the pictures, we could see T cells were specific for tumor in this case, but they weren't able to get to a lot of the tumor. The tumor had formed these really massive cyst-like structures structures that didn't allow for good, proper presentation of antigen. We saw T cells getting rid of a lot of the loosely associated cells, but not able to get into that. And so a lot of researchers are looking at making tumors, not just treating tumors with tumor-specific T cells, but then also trying to create, make tumors express antigen better and making them more accessible for lymphocytes. So those are treatments people are looking at in conjunction with T cell therapies. So regulatory T cell responses, memory T cell responses,